will recap. Okay, so far, to draw a good table, number one, you should have title with units. Number two, you should have a table. Your table organization should be well done. Independent variable should be at the leftmost column. Everything else, the rightmost column. So right column onwards. You should collect the data preferably downwards. Don't let your data extend horizontally across the page. Okay, and you should have headers with units. Last but not least, your data, you should have a complete table. What do I mean by that? If you're asked to conduct an experiment with 5 different variables, you should collect all the data possible. Sorry, that's not the last. The last one is a trend. Yeah, okay, thank you. Okay. Your results should show some trend. It should. Three of these points are within your control. You can include one more section, complete with correct precision of your data. Three of these points are within your control: title, table organization, and you collect all the data possible. It's the last one that's out of your control. Okay, but what does this mean? When it comes to a practical, really we just want to throw some marks into your face. Right? Just collect all the redundant stuff. It's tedious, but if you get it right and you're meticulous, we'll reward you for being meticulous and neat and organized. The trend of the data is the one that everyone's trend may be slightly different. Right? But it's just one mark. Right? But everything else is within your grasp. Today we're just going to practice 1, 2, and 3. Those that are done, may I get you in your OneNote to try to open this particular link on top? Let exchange. Okay. In order to do that, I think you have to scroll all the way up to OneNote. You will see the icon of your document. If you double click that, you should be able to click on preview. Preview should bring you to a page that allows you to click on the link. Okay, this is what you should see. Okay, I'm sure some of you have begun clicking, exploring the simulation. Right now, we're going to practice drawing a table, collecting data, or learning at the same time. We're going to complete the chapter by learning about the factors that affect photosynthesis. Here in this simulation, and you can see on the screen, you see a big yellow box and a blue box below. In these boxes, graphs will be generated in a while. How do you generate a graph? If you look at the screen, there's run, there's live, there's reset. Every time you press run, a graph will generate to show you the results of the experiment. Okay, we're going to show you an example. If you are on a simulation now, may I get you to click on glucose concentration, and then the box will turn pink. We are going to use glucose concentration as a measure of the rate of photosynthesis. Photosynthesis produces glucose, right? So we'll use that as a measure of how fast photosynthesis is going. The less glucose, the slower the rate of photosynthesis. The more, the faster the rate of photosynthesis. Once you click on glucose concentration, you'll find that to the left-hand side, there are many factors that you can adjust. There is the pH of the stroma. Oh, I just did something weird. Okay. There is NAD pH concentration. There's carbon dioxide. There's temperature. And then there's atrazine, which I have no idea what it is either. You scroll up, you also see distance from the lamp or the light. Today we're just going to investigate how 3 factors affect photosynthesis. This is what is required in your scope of learning. Number 1, distance from light. Number 2, carbon dioxide concentration. Number 3, temperature. It is required of you to understand how these 3 factors affect photosynthesis. Right now, as individuals, I'd like you to just choose one factor you'd like to investigate. Distance from light, or carbon dioxide concentration, or temperature. Okay, mentally decide on one right now. Later on, we'll share the data that we collected as a class. Everybody will collect different data. Yeah? As with every experiment, you must know what variables you want to control, which variables you want to keep a constant, yeah? And which ones you will change. So, if you are going to investigate how carbon dioxide concentration affects rate of photosynthesis, you don't adjust anything else. Okay? If you want to investigate how temperature affects rate of photosynthesis, you only change the temperature, but you don't change any other factor. Right? That would allow you to investigate how the factor affects. I would like you, as individuals, to choose 5 different points to change. For example, if you are working on temperature, you run the experiment 5 times using different temperatures, 5 different temperatures, so that you can investigate and do this experiment yourself. If you choose carbon dioxide concentration, keep everything else constant, but you choose 5 different concentrations of carbon dioxide to play with. I'm going to show you an example now. Let me adjust temperature. Okay, let me just work with 25 degrees first. I'm going to click run, and this is what you'll see generate. Ignore the graph on top. The graph on top shows you a lot of information. Focus on the graph below. If you look at the graph below, I would like the data to collect is the amount of glucose generated after 40 minutes. So I want you to look at the final line. So how much glucose has been generated here? 2 micromoles. If I change the temperature to, let's say, the next temperature, I want it to be 38. Okay, maybe since we go at 25, let's go to 50 next. Then I click run. You will see a set code. The plant is dying. Let me go one temperature back. I think that was too high for the plant, it died. Let's go at 40 degrees, let's see if it's still alive. Alright, still alive, okay. So here I have 3 data points already. I have 25 degrees, 30 degrees, 50 degrees. And I have 3 outcomes of the result. Right? Okay, so your task now is to draw a good table, collect 5 different data points. Right? And then after that we'll share the data that you collected. So may I just see who is doing distance from lab? Okay, may I see who is doing concentration of carbon dioxide? Last but not least, who is doing temperature? Alright, okay, you may proceed to collect the data and then we will share in a while.
Do you want us to write all this down in like a table?
You mean the data?
Yeah, like this from the graph. You need us to like write it.
Oh yes, so that's what we're practicing now. I would like you to draw a table to collect the data based on what factor you are changing. I'll show you a sample of the table. I'll leave it up here again. You modify the title and the headings based on what you are collecting. Okay, we give you a practice, so prepare you for when we eventually go into the lab. Now invite 3 people up to draw their tables and data. therefore there's still some control. doesn't change after a certain point. So if it's low, it's at 0, right? After you go higher, it just stops at 0. Alright, good one. If you are done and you are free, nothing to do, you could read up on the next set of notes, give it a browse. Do you know how to use a microscope? When's the last time you used a microscope? JH1? Oh, bless. Seldom. Okay, so you are not very good at using microscope. Or maybe experts, huh? Okay, good. Is it also the same? You are not bad, huh? Your right so straight. No, like Paris is so neat. Okay, thank you, three of you. Let's have a look at their tables. First and foremost, we look at their titles. Everyone has a title. The titles also come with units. Okay. Next, we look at the organization. Independent variable flush to the left. All of them also come with units. Your dependent variable flush to the right also comes with it. Okay. So this one, I think is missing a unit. Okay. This one has units. Okay, then is the data complete? Yes, they completed it. Right, they collected as much data as they could. Is there some sort of a trend? Okay, this is where we are going to talk about the factors that affect photosynthesis. First and foremost, let's look at distance from the lamp. You notice that when the lamp is very close, the rate of photosynthesis is pretty high. As it goes further away, it decreases. Very far away, it's not going to, rate of photosynthesis is at its lowest. No more glucose is produced. Right? Okay, so the trend we see so far is that as the distance from the light increases, the rate of photosynthesis also decreases. Right? Based on what we see by the amount of glucose formed. Now the question is why? Which stage of photosynthesis requires light? The light-dependent reaction, right? The first stage, right? So when we do an investigation on the distance of lamp, what we're really investigating is how the amount of light affects the first stage of photosynthesis. There's more light, you're likely going to have a higher rate of photosynthesis. Okay, so which is the stage that's really affected? It is the light dependent reactions. This stage is affected. The less light there is, you can expect that less electrons will be excited, less ATP, NADPH is produced for the next step, the Kelvin cycle. Okay, so that's how you can relate that. Okay, light dependent reaction. Okay, some words you could use. You could bring in how electron excitation is affected. You can also bring in the idea that less ATP and NADPH will be produced. Once you are done, then you think about this. Which stage is CO2 used in? What is this experiment really investigating? Is it the first step or is it the second step? Then CO2 really affects. Okay, I'll pause a while now. I have you until 2 o'clock. What about CO2? We notice that the more CO2 you give, actually up to a certain point it makes no difference anymore. Perhaps the first point is, where is CO2 used? It is used in the second step of photosynthesis, the Kelvin cycle. But we find that after a certain point, it makes no difference how much more CO2 you give. Why is it so? See, at the end of the day, first step and second step, they are all run by enzymes. You have a limited number of enzymes. You can give more CO2, it won't make a difference if the enzyme can only carry out reaction at that rate. Okay? So you are limited by a lot of things. Number of enzymes, it's just one example of something that you are limited by. You can give all the CO2 you have in the world, but it won't make much of a difference unless you give it more machinery to work with. I actually cannot tell if you are still writing or not. Until I peek over, I realized a lot of you are doodling. Okay. Last but not least, temperature. Every time I see temperature, right, it relates to one thing we've learned before. Proteins. Remember how proteins and enzymes are affected by temperature? At the end of the day, remember, photosynthesis is also run by enzymes. At low temperature, okay, so when temperature is low, you can expect that the amount of kinetic energy is low. So the rate of effective collision between your enzyme and substrate will also be lower, and then the reaction runs slower. But as temperature increases in this region here, as temperature increases, you find that the amount of kinetic energy also increases, the rate of collision also increases, more enzyme substrate complex can be formed, and your reaction proceeds faster. That begs the question, what happens here? How come when temperature is so high, things take a turn? Okay, so if we put it very simply, the enzymes denature at very high temperatures. What's the point of showing you this? Okay, again, even though we are starting to move away from the small things, everything still is grounded in your basics. Okay, if your basics are strong, biology is just a build-up from what you've learned so far. Okay, for those that are done, please drink some water, refresh yourself. The temperature these days is getting hotter and hotter. I read the news night time 29.5 degrees, close to 30 degrees at night. Wow. If you like to, you can take a photo of the data your friend has collected, so that you have some reference and something to base what we are discussing off on. Once you're done, drink some water. Need a sweet from the snack corner, please grab some. Hydrate yourself, get some sugar. By fast money. So essentially you all pay taxes. Anyone want sweets? Anyone wants? Any catch? Go. Hiya. Oh yeah. Anyone else? Right, last two topics of the year. And bye-bye already, your JH4. So fast. Okay, officially, we are going to leave behind the small things that we've been studying, from biomolecules to cells. Because right now, we're going to use those cells to build bigger things. Okay, we're going to use cells to build bigger things. And we're going to look at systems. Before we dive into what systems are, I'd like to hear from you. What are systems to you? Are there examples of systems in your life other than the ones you heard of in biology? And what are some features of these systems that make them systems? Just very quickly, a 2-minute buzz with your neighbor. You don't really need to write anything down for here. It's just an introduction to the topic, right? Just to kick things off. Okay, so very quickly, we just discussed with your partner some systems that you know of. They are outside of biology. Okay, what makes a system a system? Okay, so I'll give you a quick 2 minutes to buzz with your partner for question 1 and 2.
Oh my god. Not necessarily. It's like a small effect. Why is it all at times?
Have you learned this in LA before? Right. Okay, systems. What are some systems you know of in your life?
Okay.
Oh, sorry. Santosh?
A pen.
Your what?
Pen.
Your hand?
Pen. P-N.
P-hand. Okay, the fact that you say pen leads me to think that you learned this in LA. Do you? Okay, because I just came from 303, the first person response is also a pen. So next thing you're going to say car. Okay, pen. Okay, please enlighten me. What is a pen system?
All the parts of a pen should work properly in order to make a system.
Okay, so say again. The pen.
All the parts of a pen.
Do you all memorize this instead of pen? Was it? Okay, you're saying there are parts that
Should work properly and orderly.
work properly. Wow. I feel like Santosh talking. Work properly and orderly together. Anything else?
In order to make a system.
Okay, in order to make a system. Okay, a pen. Wow. Okay. Anything else? Any other systems you know of? Okay, Ishan says the education system. What makes this a system?
It's not the fault of the student, it's the fault of the education.
Are you okay? Okay, please explain to me this fault part.
But I don't know that last year that when they taught systems in LA, they talk about the input. And the input are normal students. Like the output is the best student.
What you learn in LA? Input, output. When we look at systems, there are inputs and outputs. Okay, fair enough. Is that what's the input for pen? Ink. Output. Oh yeah, okay, maybe an essay. Okay. Any other systems you learned of? Or you know of? They think a system. PA system. What makes the PA system a system?
It doesn't even work properly.
Okay, does it work properly? Okay, anyone from PA here? That don't insult you. Okay, PA system. Okay, okay, about whether it works properly or not. Any other systems? We will take one more. Wait. Sorry? Government. Okay, the government use also a system. What makes the government a system?
All the ministers to work.
Okay, you look at all the ministries. Yeah, all the ministries. Many parts that make up the government. You were saying ministers also. And all the ministers that make up the people in the parliament all coming together to make up the government. Your understanding of what systems are won't vary that much from what we're going to learn in biology. When you look at systems, you also broadly look at a few things. First and foremost, when we look at systems, we want to know their parts. Okay? Okay, so last two topics of the year are systems. We will usually start off with the parts. What are the parts of the system? It is an introduction. I will not ask you on this. I want to ask you what a system is. I'll leave it to LA to do this. Okay, so what are the parts? That's usually how each topic will unfold. After we know the parts, we will then try to figure out how they work together. Okay, that's usually the second part of each lesson. After we know how they work together, usually one of the things we'll look at next, to end off the lesson, is to look at how it can be disrupted. For example, we're going to learn about circulatory system. What if one part fails? What if the vein, artery, or the capillaries fail? What if your heart fails? How does it affect the parts working together? So typically, when we study systems in biology, you will see three broad areas: the parts, how they work together, and how they disrupt each other, how it can be disrupted. I would say the parts are usually a little bit more factual. That's where you gain a little bit more vocabulary, so that you can describe each part properly. Usually, when we look at parts, certain skills come into mind. We want you to be able to identify. If I give you a heart, I give you the organ right in front of you to dissect, are you able to identify the various parts? If I give you a plant and you were to slice it into under the microscope, will you be able to identify the various parts? When we look at how things work properly with each other, you will normally see us asking you to describe how it works together. Okay, so that was just an introduction. For us, we're just going to end up with two systems, the transport system in plants and in humans. With that in mind, I'd like to see how much you know from your primary school days. I believe you have learned a little bit of transport system in primary school, from plants to humans. I'd like you now, in your pairs or triplets, to have a quick buzz. What are the similarities and differences between the transport systems in plants and in humans? Okay, you can write it down in bullet points, only in full sentence, so they can have a quick discussion. Okay, we'll wrap up your discussions. Okay, no chance I'll be doing, you'll see this in exam, okay? But we want to use this as a launch pad so that we see some commonalities as we study these two systems. First and foremost, do you see any similarities between these two systems? Yes, you're about to say something? Yes. Yes, Santosh?
The nutrients are close to all parts of the body.
Is that the similarity?
Yeah, all parts of the system.
Okay, so in both systems, you are transporting.
The nutrients are transported to all parts of the body.
Okay, so in both cases, you are transporting nutrients to all parts.
Isn't true.
Is that true? It is true. In plants, the food that's made in the leaves will be transported to every part of the plant. In your human body, nutrients will be transported to every part. Okay, that is true. Any other similarities between the two systems? Zachi, you have any similarities?
The one I thought is the same as transporting nutrients.
Okay. You have which one you contribute? Any small similarities or differences?
Like one is sort of directional, while the other is like both are directional.
Both are?
Directional.
What do you mean by directional?
Like, the Xylem only goes one way.
Okay, Xylem only goes one way, Phloem goes another way. Oh, okay, there's no like backflow lah. Okay, your blood will flow in one direction. Okay, there is some directionality in both cases. If you look at the plants, you'll find that water is always channeled upwards, from the roots to the leaves. I was told that in primary school you learned this word called transpiration, which is the evaporation from the leaves. And that is what results in water always going from the roots to the leaves. And food will always be transported from the leaves to all parts of the plant. When we look at humans, however, there's also some sort of a direction. Do you learn that in arteries are the ones that carry blood away from the heart? And then veins are the ones that bring blood back to the heart? Okay, some of you did, some didn't. Okay, so there is some directionality also in humans. Any similarities or difference? There's one big difference. Okay, there's a pump lah. If you ever come upon a plant and you see what you don't have, right? Okay, so a pump. It's a part I find very fascinating. When you look at animals, we rely on a pump to circulate things. But if you were to cut down a tree, you see no pump. Yet the trees can go at least 3 or 4 times, 10 times taller than a human being. How is a tree able to transport things to every part of its body without a pump? Okay, then you all learn about transpiration, right? Okay, so we are already building upon what you learned in primary school. Any other similarities or differences? Yes.
Me and mother were talking about how like in the plant there's it only transport the food and water and that's it. But then like in the human body you transport oxygen and the cells need to like use that to produce energy and everything. So basically like the plant is what is being transported is really like really used directly. But then in the human we still need to go through all those reactions.
Oh, okay. So are you saying that in humans there are a lot more reactions take place?
Before like the final product is produced, yeah.
Okay, but for plants,
It's just direct, the food already comes direct. Don't need to do any reaction with that food. The glucose is there already.
oh, okay. Okay, then the topic we just studied, photosynthesis, actually comes with many reactions also. I think maybe it's the part, on our part we watered it down a lot. Yes, actually plants do have a lot of reactions to get to whatever products it have. For example, glucose, we simplified a lot of processes, so you have less to remember. Okay, but I'm glad you brought up this point because indeed a lot of reactions take place for these products to be produced. But if anything, once these things are produced, they need to be transported to the right places also. Okay, in both cases. Perhaps one similarity are the tubes. Yeah, in both cases they are tubes, right? Oh, sorry, maybe vessels are better word. Not so much tubes. In both cases, they are vessels of some sort. In plants, they are vessels. In humans, they are also vessels. They are like runways in the body that allow for fast track movement. Or they are like the highways. Right, okay, then we look at plants, right? There seems to be only two kinds of vessels, the xylem, the phloem. Whereas in humans, we seem to have more. I think one difference that you know of, but was not brought up, is what is transported. You all in primary school? Xylem and phloem, they transport different things? Yeah? So that is another area that we're going to look at. For today, in the remaining time, we're just going to look at the vessels. This looks at the parts. You want to understand the transport system in plants well, you've got to look at the vessels. When we go to human transport system, we'll look at vessels and the pump. Luckily, plants do not have this pump, so we only have the vessels to look at. It's quite disturbing, right? You can't open a carrot and see one beating heart there. Okay? Right now, we're going to dive a little bit into the structure of the xylem and phloem. You've learned a little bit about xylem and phloem before, so take this as an extension of what you already know. May I get you now to go to your textbook? We're going to do a quick read of the chapter. There are 2 pages only to describe the xylem and phloem. May I get you first to focus on page 2? Okay, I'm going to go through this exercise. There's a lot of vocabulary that's very new to you. May I get you to read through page 2? Highlight any words that confuses you or hinder your understanding, and we'll run through the vocab very quickly. Okay, just a quick read of page 2, 3 paragraphs. Highlight all the vocab you do not understand, and then we'll run through them on the board. If you're done reading, just shout out some vocabulary that hinders your understanding of the chapter, of the page. Okay, cellulose, microfibril. Thank you. Sorry? Perforation. Okay. That's very wrong. Oh, is it O or T? O. Yes, it was intended to be a O. Anything else? Ooh, that's not. Okay, so primary versus secondary cell wall. Nico? What? What's a paper? Oh, tapered. Tapered. Okay, anything else? Yeah, I just need to read page 2. Then we'll have a look at what this vocabulary means. page 2?
They add maturation.
Oh, what does date and maturation mean? Okay, that point. What does date and maturation? Oh, it sounds like very sad life, huh? It's like you reach adulthood and you die. Date and maturation. Okay, let's have a look at the vocabulary for this page so you can understand what the xylem is. In primary school, you learn about the xylem and you learn that it transports water. That's what the first word over there says. Right? It says water conducting cells. Conducting means to conduct, means to get things moving through it. Like how something can conduct electricity, it can conduct water. Now let's overlay the information in a JC level. Actually, there are two types of xylem. Okay, we have tracheids as well as vessel elements. So these are the two different kinds of xylem we would like you to know from now onwards. There are some vocabulary that you are encountering for the first time. First, paragraph says these two types of cells are dead and maturing. Wait, the English very odd. Okay, it's dead and maturing and contains no cytoplasm. What does that mean? It means that every cell goes through a life cycle of sorts. They start off as a young cell, they can mature into a matured cell, and they will eventually die. What this sentence is saying is that when these cells mature into its final form, it will after that it will just die quite immediately. Okay, that's what it means by dead and maturation. There are other cells, most cells in our body, once it's mature, will continue to stay alive. Right? So that's the point it's trying to make. Dead and maturation. Now let's have a look at the two kinds of xylem that you encounter. The skill, yes.
How does it get transported and there's no pump and it's already dead and maturation? How does that like transport water?
Right. How does it transport water if it's dead? Is that your question?
Yeah, like how does it move?
There's no pump, is it?
There's no pump and it's already like this.
Right. Okay, so that's a bad point. There is no pump. So how can water be transported, right? That's why next week we will learn about the processes that allow water to be transported even without a pump.
Is it a transpiration?
Yeah, and one of them you learn in primary school, transpiration, the evaporation effect. Okay, so two kinds of xylem I would like you to know. First one is tracheids. Second, vessel elements. But what is the real skill to pick up here is to be able to identify them. If I were to provide you an image like that, will you be able to identify it as a tracheid? If I provide you an image like that, will you be able to identify it as a vessel element? Okay? Later on, we'll practice before we go. Okay, so let's go on with the vocabulary. It says tracheids are long slender cells with tapered ends. Here is a cell without tapered ends. Here is a cell with tapered ends. Okay, tapered just means towards the end, it connects and joins to a point. That's what tapered means. Okay? If you look at the image inside your textbook, you can see just that the cells have tapered ends. They connect to a single point at both ends. Can you see it? Like look at one cell over here, it tapers down to a point. Here's another cell that tapers up to a point. Okay, that's what tapered ends mean. Not so much for you to be able to describe, but if I provide you an image, you are able to identify. Okay, our class did not ask what pits are. Okay, these pits aren't armpits, but the idea is the same. It's an indentation, they are holes. Tracheids have many pits. These are holes that can be found along the cell.
Is that where the water comes out from?
Yeah, so imagine lots of tracheids. Okay, let me align lots of tracheids. Lots of tracheids. Not only is water traveling from cell to cell like a straw, if it encounters a cell next to it that requires water, some water can exit the pits to go to the cells.
What does it flow up?
Sorry?
It just flow up.
Why does it just like that? Okay, my only conclusion for this, right, would be that the forces driving up is stronger than the forces driving out to the sides. Then you may be wondering then what forces will drive out to the sides? It really depends on how much water your cell needs, right? And then you learn about osmosis. Yeah. So I think it will depend on how much water the cell needs at the site. Okay, so these are tracheids, huh? You expect you to be able to identify them. The next kind of xylem that you encounter are called vessel elements. Vessel elements are a bit cuter, they're a bit shorter. So if I were to draw a vessel element, they are broader, but each cell is much shorter. They also come with pits, exit points for water to reach surrounding cells or tissues. But not only that, they come with, okay, you find this word in your textbook called perforations. Perforations are also holes, but they're more like slits. Okay, these are perforations. One look at this doesn't quite make any sense. How can this transport anything? Well, we need to look at it from a broader perspective. Imagine if I have many of these cells stacked upon each other. Then suddenly I've got a tube. Okay. Suddenly I've got a tube. A tube that has many perforations allows water to move up from the roots, eventually to other parts of the plant. Okay, so we've talked about what tapered is, we've talked about perforation is. Let's talk about these two points here. What are primary and secondary cell wall? What is cellulose microfibril? Let's talk about cell wall first. You know that plant cells come in cell wall, right? Okay, so I'm going to draw a cell wall right now. Plant cells have cell membrane, which are made out of which biomolecule? Okay, lipids up. Okay, let's link back. Next, plant cells also come with cell wall. What biomolecule is this made up of? Sorry? Cellulose, yeah, which is a carbohydrate. Yeah. Okay, so cell wall is made up of cellulose. Okay, so let me just relate it up here. Cell wall is made up of cellulose. Okay, that's what the cell wall is made up of. So what is the word microfibril? Micro means small, fibril means fiber. So what they are saying is, your cell wall is made up of many cellulose fibers. And that's what it means. Last but not least, then what is a secondary cell wall? All xylems come with not just the usual cell wall you know of, it comes with an additional layer of cell wall. This makes the straw, the tube, very strong and sturdy. It won't just collapse. That's how trees are able to transport water away from the roots all the way to the top of the very leaves. These tubes need to be reinforced. So, the interesting thing is that secondary cell wall is not found on the outside of primary cell wall, it's found at the inside, sandwiched between the cell membrane and the primary cell wall. Okay, so what do I mean? Right now, if the red one is the cell membrane, the black one is the primary cell wall, plants make new cell wall from the inside out. Okay, so imagine I'm a plant cell right now, using purple to represent a new cell wall being made. It will make new cell wall here. As the new cell wall is being made and it thickens, it will push the old primary cell wall upwards. Okay, so it grows from inside out. And that's why if you look in your textbook and you look at this image over here, you'll find that the primary cell wall is on the outside, secondary in the middle, followed by the cell membrane. Okay, it's because of the way the plant cell grows. Any questions for now? One last point, then we'll end off today. Okay, the beauty of the secondary cell wall is that it contains a very special carbohydrate called lignin. Lignin is a carbohydrate that we did not cover in our syllabus. Carbohydrates you know of are starch, cellulose, but actually there's a third kind of carbohydrate that's found in a lot of organisms. It's lignin. It is what you find in mushrooms. It is what you find in insects. Okay? Yeah? You know how insects' exoskeleton is very tough? Actually, it's made of carbohydrates. It's made of lignin. Lignin is found in the secondary cell wall. And very often, if you get the privilege of looking under the microscope, maybe next week, I want you to spot how lignin is deposited in the secondary cell wall. Sometimes it's deposited in a very spiral fashion. So imagine the lignin isn't just, patched up all the way. It is often deposited in a very spiral fashion, this lignin. And so usually one way to spot a xylem vessel is by spotting the deposition of lignin. If you see lignin deposited, very often it's a sign that this is a xylem vessel. Because it needs to be reinforced, it needs to be very sturdy to be able to transport water. It is an epically long straw. And so let me show you an example of how I will assess your understanding of this part of the chapter. If you look at CA 4.1, sometimes we'll provide you a microscope image. And then we'll ask you to identify where the tracheids are and where the vessel elements are. One look over here, can you tell where the xylem is? Can you where the xylem, all the parts of the xylem are? Can you tell which one are tracheids, which one are vessel elements? Okay, before we go today, I'll just get you to identify these two things. Okay, could you label what you think is a trachea? Label what you think are many vessel elements stacked upon each other. Okay, so let's close off the day. Xylem, is it on the left side or the right side? Left side. Like this side over here? Okay, so one clear sign of whether something is a xylem usually is the deposition of lignin. So you try to spot something that is deposited in this spiral-like fashion. Actually, the lignin has been stained here. It's been stained pink. Can you see the lignin has been deposited? Yeah. Yeah. All these pink lines, they represent the lignin spiraling upwards.
Then what about the other like one?
This one over here?
Yeah, one also got the.
Yeah, you see, it's been deposited with lignin, and then like somebody like erased the center away. Okay, I suspect it's because of the way this specimen was sliced. It was sliced in a way that this tube was just sliced in half, so you don't quite see it go all the way around through. Okay? So where are the tracheids? I would say the tracheids are like this region. You see that the cells here are rather thin. And if you look at some of the cells, they do look like they're tapering at the ends. Okay, so this region over here, lots of very thin, long cells. Likely they are the tracheids. This thick tube over here, with one white hollow channel all the way through, that's likely the vessel elements that they stack upon each other. Okay, the thicker one. But what's the moral of the story here? Vessel elements are way thicker than tracheids. Okay, and we will stop here for today.
Wait, why can't the thing on the left be the vessel elements? Yeah, no, more on the left, even more the extreme
Like
left.
here?
No, left.
Like here.
Yeah.
Like here, is it?
Yeah, isn't it like stacked?
Right, okay. So in my past experience, I would have said yes. But as I look at real specimens more and more, I find that xylem vessels tend to have lignin deposited. Lignin means it's an indication that there is secondary cell wall. Okay? Xylem compared to phloem does not have secondary cell wall. So nowadays I like to use the secondary cell wall as a sign that something is xylem. Right? Because xylem needs to be reinforced.
Everything with the red thing is all
The lignin being deposited. To give it some structural integrity.
so the rest are all the one.
Yeah, so very likely next week we'll look at phloem. Likely these are phloems. Yes. Right, okay, so these also have tapered ends, right? Okay, but again, I would say because I don't see secondary cell wall. And the sign of that usually is lignin. You'll find that when we go into phloem also has some cells that have tapered ends. Okay? Yes. Why does a phloem need lignin? Okay, I suspect it's because of why it's being transported, which are sugars. Lignin is waterproof. So it kind of keeps this straw, allows the water to stay in the straw in a sense. Yeah. So that's my suspect of, that's why I suspect why phloem does not have this lignin. Okay, so this is where we'll end off for today. Next week we'll look at the phloem. Okay, go have a short break. Then I'll see you at 2:30. For the two groups auditioning today, then you audition. Okay, but for the rest of you, at around, you're going to have the audition at 4:15. You're going to watch, then you stay, but you'll be quiet. Okay, so that means I will start to dismiss all of you from around like 4 or 5 onwards. You can be dismissed. Okay, so that you can give a quiet space for your friends to audition. Okay? But if you want to give them moral support, you can stay behind and watch also. Yeah? Okay, go for a short break. I'll see you at 2:30.
Until the last break. Until the last break.
quite on this level is they just want to have management. So they just want people quite consistent. Right, you know, remember the natural transport chain of photosynthesis, right? The electrons first react. Okay, so there's phosphate from of which so as you see the diagram, remember the nice strike and the electron come out right? Yeah, it came out from the circle. So that's how it works. Yes. Don't worry, it's not that bad. You must have a little bit of confidence. Yeah, actually you're doing well. Put yourself down.